Good morning, everyone. Welcome to Fast Forward in 25. I would like to introduce our guest speaker for today, Mr. Mannan Khambati, Vice President, Bharat Serums and Vaccines Limited. Currently, he leads a highly motivated team at Bharat Serums and Vaccines Limited, a Mankind Pharma subsidiary specializing in development, scale up, and manufacturing of recombinant therapeutic biosimilars, hormones, and the world's first novel antibodies. With a strong background in process improvement, GMP compliance, and biotechnology, he has held progressive leadership roles during innovation and excellence in biopharmaceutical manufacturing.
So, thank you very much for the kind introduction, and thank you very much for giving me this opportunity to speak with all the students. So, I approximately have around 60 minutes to speak about, and I'm going to talk about novel biologics, a promising future landscape. Okay, in between the presentation, if you have any questions, you are free to put them in the chat box, and maybe at the end of the session, during question and answer session, we will take them. Okay, so today's agenda, I am dividing this into three parts. First is introduction to biotechnology. I assume a lot of students are from different fields. So a small introduction of what is biotechnology, what is the market economics, and then the main focus would be on upcoming technologies. So let's introduce what is biotechnology first. Many of you probably would know what is biotechnology, but still I would just for the benefit of everybody explain a bit. Biotechnology uses living cells to develop or to manipulate products for specific purposes, such as genetically modified food. Biotechnology is thus linked to genetic engineering and emerged as a field in its own right at the beginning of the 20th century, which was later joined by other sectors such as medicine and the environment. Modern biotechnology mainly is divided into five branches targeting human, environmental, industrial, animal, and plants. Thus, help us fight hunger and diseases, produce more safely, cleanly, and efficiently, reduce our ecological footprint, and save energy. All this has excited the stock markets like Wall Street, thus making biotech one of the most profitable sectors of the Nasdaq Composite Index 2019. Biotech as a commercial option. So, biotech has been used mainly into biopharma for drug production, gene therapy, and drug testing, and agriculture, tolerance to pest, increased nutrition, novel substance production, animal husbandry, improved breeds, drug production, faster growth, and environment, bioremediation, and biodegradation. But here, mainly, I will be talking more on the biopharma because I belong to biopharma. And in biopharma, I mainly do drug production. So, this is a typical evolution of biotechnology starting way back in 1919. An Hungarian agronomist, Karl Erke, coins the term biotechnology, and then in 1928, the famous case I think everybody must have read in the textbooks about the Scottish bacteriologist and Alexander Fleming, who discovered the antibiotics effect of penicillin. In 1943, we have Oswald Theodore Avery, who discovered DNA as a carrier of the gene. And exactly after 10 years, James Watson and Francis Crick described the double helix of DNA. In 1969, the enzyme is synthesized in vitro, and 1983, the first genetically modified transgenic plant is presented. 1997, the very famous case of Dolly being cloned as the first mammal, and 1998, the human genome was mapped, around 30,000 genes were located. Year 2010, we have the first synthetic cell, and 2013, we have the first bionic eye. And of course, everybody present here would be knowing about year 2020 and the discovery and fight against the COVID virus. So, biopharma medical technology is again divided into various sectors, as I have mentioned earlier. We are talking about recombinant pharmaceuticals, we are talking about stem cell therapies, gene therapies, diagnostics, tissue engineering, antibody therapy, DNA fingerprinting, human genome project. Again, I will be more focusing on the recombinant pharmaceutical, otherwise, it is not possible to finish this even in 2 days. So, commercial products from biopharma, a few examples. We have recombinant hormones, a very famous example of insulin. I think many of the households use today the injections. the growth hormones and follicle stimulating hormones. We have blood products like albumin, which are naturally produced in blood, but now people who have deficit of those are being produced via biotechnology and genetically engineered cells. Same applies to various types of cytokines like interferons, interleukins, and of course monoclonal antibodies mainly being used for treatment of cancer in oncology products. You have various varieties of them being produced today, and we have recombinant vaccines like recombinant peptides, DNA plasmids, etc., and recombinant enzymes, miscellaneous products, and personalized therapies like gene, cell, and tissue therapy. We will discuss more about them a little bit in the later sector. So, a typical biotech process flow looks like this. It has 4 stages. It has development stage, which starts with the gene manipulation, the genetic engineering, choice of host cell, which can be bacteria, yeast, or mammalian, which depends upon the type of protein that needs to be produced. Once we have a genetically stable host cell, then the master cell bank is made, which is frozen in liquid nitrogen so that it becomes immortal. From master cell banks, we produce working cell banks, and from working cell banks, we start the second step of upstream. Here, the cells are put in media, particular growth media and environment is given so the cells grow. And then the cells are scaled up so that the volumes increase. Because of the volumes, the cell quantity would increase, and because of the cell quantity, the amount of protein they produce will also increase. So then it is taken into a stirred tank bioreactor, or different types of bioreactors are there. Here the cells grow and they produce more amount of protein. The protein can be either inside the cell or outside the cell. In whatever case, the protein is of interest. So here we harvest the cells, separate them, and separate them from the liquid which contains the protein. This goes to the third sector of downstream. In downstream, again, we will do various types of chromatographies to do purification, and then some polishing chromatography, and finally concentration, so that we will have a purified bulk which can be frozen or it can be put into cool temperature and used as and when required. When the bulk is required to be formulated to make the final injection, it moves into the fourth stage, which is fill and finish. So here the bulk is taken and formulated to make it more stable. Various tests are done, and then it is stored, and eventually it is put into the injection formats. Per se, right now, polishing is to remove other ingredients which should not be present in the bulk, because we want the protein to be 99.9% pure, because ultimately it is going to go into humans. So there are various chromatography steps for removing different types of impurities. One of them is polishing. Okay, yeah. So a little bit, very small sector I'm taking about the economics. Why we are doing this is because, according to various research reports, the global biologics market size is expected to grow between 500 to 800 billion. This slide is a little older, so I think this 800 billion must have converted into a bigger number by now. But anyway, that keeps changing. So this is the basic picture of why we are doing this whole thing. Is by year 2030 and a CARG growth of 4 to 6%. Biologics market is growing steadily because companies are spending a lot of money to improve the research and manufacturing facility. In-house R&D and manufacturing are still present in-house, but then contract manufacturing, contract research is increasing simultaneously, because everybody is not able to produce so much in-house that they have to do contract manufacturing. So there is some regulators role also. I'm just bringing a different angle to this because you must be hearing a lot of US FDA inspections happening, a lot of companies having findings in the US FDA. Now this FDA findings are a problem statement, but they also create an opportunity for the companies. Why they create opportunities? Because it makes the company better. It also makes the technology better, because now we are able to bring in new technologies like single-use technologies or modular technologies or continuous processing, which actually helps the industry to update its old method and reduce the risk that comes with making the big changes to existing systems. Now, the main focus area of upcoming technologies, I think I will spend more time over here. I am just claiming that I am not an expert in all these technologies. I am mainly responsible for recombinant regular manufacturing of hormones and MABs. But here we can discuss more on the science part of it by giving few examples on how the stem cell, gene, and tissue therapies, which are more personalized therapies, are being used. the DNA-based therapies, bispecific antibodies, antibody drug conjugates, FC fusion proteins, IgM antibodies, and other alternatives, engineered exosomes, mitochondrial engineering, pharmacogenomics, AI and machine learning, and plant-based biologics. AI and machine learning is again a very generally used term. How we use it in biotech is something that is very interesting, and I will explain that in a little bit. So, first, to speak about gene and cell therapy, this is also quite in trend right now. Of course, in countries other than India, this has actually come into market, and India also a lot of companies are doing work on this. So, in gene therapy, use of adeno-associated viral vectors are done to deliver therapeutic genes, and they have shown promising results in treating genetic disorders like Parkinson disease, spinal muscular atrophy, corneal and retinal repair, and hemophilia. Additionally, cell therapies like CAR T cell therapy have demonstrated remarkable success in treating certain types of leukemia and lymphoma. Just explaining in a bit how this gene therapy process flow works. If you can see this diagram, usually the cells are removed from the patient. In the laboratory, virus is altered so that it cannot reproduce. A gene is inserted into the virus. The altered virus is put into the cells of the patient. The cells which are now transformed by this genetically manipulated virus is put back into the humans. The altered cells are injected and then they start growing, and when they grow, they produce the protein of interest, which was earlier absent in the human. Cell therapy has almost similar kind of flowchart. To talk about CAR T therapy, I think CAR T cell therapy is mainly in highlight today. One Indian company coming from IIT has actually put this into a commercial use and have demonstrated and also demonstrated how the cost of the therapy can be reduced. A few years back, one case had come where a girl had to be injected the CAR T therapy. One injection was costing somewhere around 16 crores in Indian rupees. Now, it is not possible for even a very, very rich person to do that, right? Now, this company in India has come out with a therapy which has reduced the cost to 30 to 40 lakh rupees. So that's a huge gap that has been done, but of course 30-40 lakhs is also not affordable for everyone. So here, mainly in India, this is also our aim and ambition to reduce the price so that it comes to general people. Okay, so in CAR T cell, normally what is done is the tumor cells are taken and the T cells are also taken. The T cells are genetically manipulated and they are activated by exposing them to the cancer fragments outside the cell. These T cells are then grown in numbers outside the body, and then the grown numbers of those genetically modified and activated T cells are put back into human. These T cells are now targeting that particular cancer and it goes and destroys the cancer completely. This is both are the personalized therapies. They have to be done with the cells of the same human. Okay, and that's why it is very expensive because we cannot mass produce this. Okay, so tissue therapy is similar. Tissue therapy is more on the constructive side of how artificial skin, cartilages, etc. can be produced. The few therapies are actually approved by FDA. This is more on the tissue engineering part, which aims to generate autologous tissue grafts with an engineering approach. Three major components are involved in tissue engineering process, including seed cells, scaffold material, and tissue formation environment is required. In stem cell therapy is quite interesting. And stem cells, you know what are stem cells? Stem cells are taken mainly from bone marrow, but they also can be present in other areas. And the stem cells can convert into different types of cells, like they can convert into liver cells or blood cells or cardiac cells or nerve cells, depending upon which area they are inoculated in, how they are grown, and what environment is given to them. A classical case of HIV being cured in 2019 because of a genetically modified stem cell put into the person, and four more people have been treated so far after that. Recently, a case of cure for type 2 diabetes has also come in, where stem cells have been converted into genetically modified pancreatic cells. They have been put into the pancreas of the patient, and the patient has been completely cured of the diabetes. And this is at clinical stage, but definitely going to come a long way. Main target here is to remove the stem cells from the person. mRNA-based therapeutics here, I think everybody knows the case of what mRNA vaccine. I think everybody present in this presentation is having that in their body. We have all taken the mRNA vaccines where we have used for COVID-19, and there is a huge potential for other diseases also. How it works: the mRNA vaccine is taken, and then it is the mRNA goes inside the cell. It produces the spike proteins of the virus, and it is exposed on the surface. The B cells get activated because of this antigen exposure and starts producing antibodies. The antibodies cover the virus, and now the virus is not able to latch to the host cell and go inside, and they get exposed to lymphocytes, and that's how they get destroyed by our own immune system. So it is a false pretense that the antigen are getting exposed on our own cell surfaces without the virus actually infecting. There are a lot of other techniques like advanced antibody engineering, where various techniques of bispecific antibodies which simultaneously targets two different types of antigen, and which has like antigen drug conjugate, and it delivers a cytotoxic drug load in the cancer cells directly. So it can attach, one arm of the antibody can attach, and another arm can attach to the cancer cell, which makes it very specific. So if you know that, like in chemotherapy, when you give a chemotherapy, it is non-specific. It goes and attacks various growing cells in the body. In this case, now the drug load can be specifically delivered to the cancer cells, because the antibody, which is bound to the drug, will go and bind only to the cancer cells. Okay, and there are certain papers also published where US FDA has approved the first bispecific antibody for treatment in adults of relapsed or refractory diffuse large B cell lymphoma, a type of cancer. Of course, everybody knows about CRISPR technology. A lot of discussions, a lot of papers are being published nowadays of this. Some ethical issues around this are also coming up, of how we can change. And a lot of, I think, movies also have been made on this. So I will not discuss more on this. Basically, it is how you cleave your DNA and remove the target part and add another DNA, which changes your DNA structure, and that can lead to different kind of reactions. So if you have a mutation in your gene, the gene can be removed and a good gene can be put in. This is in a good way. In movies, they show how monsters can be created by this. So I'll leave that. We have this novel target and pathways, like researchers are continuously identifying and validating new targets and pathways for novel biologics. This includes targeting specific receptors, signaling pathways, and immune checkpoints to modulate immune responses, regulate cell functions, and combat diseases like cancer, autoimmune disorders, and inflammatory conditions. We have biconjugates and fusion proteins. I have spoken a bit about this in past, so I'm not going to repeat the whole thing. It is almost similar. It is only differences how it is being done and for what purpose. And the next generation vaccines. Here, the research are exploring the one next generation vaccine is mRNA, but other than mRNA, we have viral vector-based vaccines, DNA vaccines, nanoparticle-based vaccines to enhance vaccine efficacy, improve immune response durability, and broaden their application beyond the infectious diseases. So here you see in between there is SARS-CoV virus, and how many types of vaccines could have actually come out. I can count the number 9 max. So you have inactivated vaccine, you have live attenuated vaccine, you have recombinant spikes, virus-like particles, DNA vaccine, RNA vaccine, inactivated adenovirus viral vaccine, replication component adenoviral vector vaccine, and non-replication component adenoviral vector vaccine. What came out was number 6, mRNA, which was the fourth runner, the fastest technology which came to market, because the platform was already ready. They just had to switch the genes. Once they came to know the genes of the sarco proteins, and they immediately transferred that to the vaccine or the host cells, and that's how the whole game began. So rest everybody working on different platforms could not make it to the market. Then we have IgM antibodies. I think everybody must be knowing the difference between IgG and IgM antibodies. Everybody produces mostly IgG antibody, typically because it is small, typically because it is easily available in the body. The main difference is IgM is very big. It has 10 sites, 10 binding sites. So, and it has a J chain that interacts with different receptors. So engineered IgM antibody represents a new class of potential therapeutics that combine multivalency, high avidity, and high specificity. They have potential in treatment of solid and hematological cancers, autoimmune inflammatory diseases, and infectious. But here there is also a problem that they're highly immunogenic. That means they can produce a very high immunogenic response in humans once given. So that's a risk here, and a lot of research is going on to remove that. Then we have exosomes. So we can engineer those exosomes. And what are exosomes? They are subgroups of extracellular vesicles which have been serving as an efficient therapeutic tool for various diseases. Engineered exosomes are sort of exosomes modified with surface decorations and internal therapeutic molecule. After appropriate modification, engineered exosomes are able to deliver anti-tumor drugs to tumor sites. So basically they are vesicles, and these vesicles become the payload. The payload is loaded with the drug, and they specifically go and pop inside the cells, and they release the material inside the cells, either kill them or modify them. Then there's a very interesting topic on nano antibodies. Now this is something new and just naturally present in camels or camel-like animals, and they are known as camelids. These are contained in the serum or as conventional heterodimeric antibodies as well as antibodies, and they are antibodies without any light chain. They are composed only of heavy chain. Okay, so if you can see in the diagram, a human antibody is approximately 150 kD. It contains these two chains on the upper side as the Y and on the lower side as the stalk. And the camel does not produce this two, it has only one VHH. Okay, so we have only heavy chain here, and this is called as HCFs or heavy chain antibodies. Variable fragments derived from these antibodies are called as VHH or nano antibodies. This since the discovery, NBS have been widely used in this field of research, diagnostic, and pharmacotherapy. Despite being approximately 1/10 the size of conventional antibody, they retain similar specificity and affinity to conventional antibodies and are much easier to clone and manipulate. The unique properties such as small size, high stability, strong antigen binding affinity, water solubility, and natural origin make them suitable for development of biopharmaceutical and nano reagents. This is typically still new, nascent. No products I think have been coming out so far as commercial at least. Maybe there are some research going on in terms of clinical. Then again, a major topic being discussed is about mitochondria. What are mitochondria? The origin of mitochondria, how they came into humans, whether they are actually an organelle or an organism which has come into it. They are called as powerhouses, right? They produce a power which is given to the cells. It's more like a symbiotic relationship we have with. But then their DNA is also responsible if, in case of mutations in mitochondrial DNA, it leads to some heritable multi-system diseases that display different tissue-specific presentations at any stage of life. Recent developments in the genetic modification of mammalian mitochondrial DNA raise the possibility of using genome editing technologies such as programmable nucleases and base editors for the treatment of hereditary mitochondrial diseases. That means that all the diseases or genetic diseases are not because of our own chromosome, but they can be also because of the mitochondrial chromosome. Now, because of the Human Genome Project, we have been able to identify which genes are present where. Earlier, we just knew about the disease. Now we know that gene present is not present in the chromosome, but present in the mitochondria. And that's how it opened the Pandora's box of the study on the mitochondria. And that's how the theory also came up that mitochondria probably is an organism in itself. And many ages back, it probably has fused with human cells and develop a symbiotic relationship where both cannot exist without each other. and it has just become a part of us. And they have their own chromosomes, which also lead to diseases. And that's why now it's on biotechnologists to treat them. This is again a very interesting subject of pharmacogenomics. Students who have studied pharmacology probably would be more aware of this. Basically, pharmacogenomics is the study of how the genetic composition of an individual affects the drug response. So if you know that every individual response to a drug is different, okay, same drug does not work. Sometimes one human has more side effects, another human does not have any side effects. This happens typically in chemotherapy. A lot of patients in chemotherapy go through a very, very bad side effects, and some patients really don't. The chemical is same, the humans are different. Okay, so the pharmacogenomics, how the humans are different because the genetic makeup is different. So pharmacogenomics, what it does, it optimizes the dosing, efficacy, and adverse drug reaction of any drug based on an individual's genetic information. It is a field aimed at understanding the genetic contribution to variability in drug efficacy and toxicity. What is the goal? The goal is to able to select the drug which is the greatest likelihood of benefit and the least likelihood of harm in individual patients based on the genetic makeup. So if I have mapped the patient, I know which type of chemotherapy or what dose of chemotherapy to be given, so there will be least amount of side effects. It is an important example of the field of precision medicine, which aims to tailor medical treatment to each person or to a group of people. So earlier we had an example of gene therapy, which was personalized. Even pharmacogenomics is personalized, individual based. And about artificial intelligence, I think you guys can tell me more about artificial intelligence than I can. What I am just highlighting here is how the AI is being used to develop drugs faster, to make the drugs available faster to humans, to study what types of drug can actually be made, how to remove the anomalies in the current drug processes which actually lead to some side effects. So a lot of things can be done, but this is based on the database. Okay, so AI and machine learning techniques are being applied to accelerate the discovery and develop of novel biologics. These technologies help in identifying potential drug candidates, predicting protein structures, optimizing drug design, and analyzing large data sets for improved understanding of disease mechanisms. So if you can see up to 22 data, there are so many drugs which have been enabled by AI and have gone into clinical phase for, so it's being used in a big way now, even in biotech. Lastly, the plant-made biologics. What are plant-made biologics? Earlier we spoke about animal cells, microorganisms, which were genetically modified and they produce the cells, but mainly the cells were grown either in suspension form or in anchorage form in bioreactors or in flasks, etc., which are present in the lab. Now, what we are talking about here is modifying the plant cells which are in plants, and of course, we need a protein which can be modified as per the eukaryotic system, and they can be manufactured in plants in a larger scale. So, plants can offer the traditional benefits of proper eukaryotic protein modification and can manufacture biologic treatments at a lower price and high scalability. Why lower price? Because plants can be grown at a very low price in larger scale. Yes, you require larger lands, but now you have this horticulture where you can actually grow plants horizontally and vertically grow both. So, saying that if the plant is modified, say for example, as a bacterial colicin example, which are derived from plants, were recently approved as effective food additive for controlling E. coli strains that cause food poisoning in food products. It costs around $1 to manufacturing 1 gram of purified choline, which makes it commercially viable. This 1 gram of purified cholines will probably form into millions of injections. Okay, and the cost is only $1. That is the production cost, of course. And numerous plants such as tobacco, carrot, alfalfa, and many more are like suitable for this purpose because they grow very fast. They're able to take the genetic modification, and it is very easy to extract the protein of interest from these plants. Okay, currently scientists are primarily primarily concentrating on areas including novel vaccine candidates, expression vectors, plant host systems, and glycol engineering for the improvement and growth of plant-derived biologics. So as I said, this is my last slide. I have finished before time. I went a little fast. These are my references that I have used mostly. And now I think we have sufficient time for question and answer. So if you have chalked down your questions, you can shoot now. How you want to do this, I leave it to the moderator. We can take one question at a time. They can ask the questions, or they want the students to individually ask them.
Hi students, if you have any questions, please ask. It's right time to clarify your doubts. And who all are having doubts, please raise your hand and unmute and talk.
All the students are very clear or did not understand. I went too fast.
Students, please raise your concerns. If you have any doubts, please ask.
Feel free to ask whatever. Don't think it's a stupid question. It can be anything.
Hello, sir. Sir, in gene and cell therapy, you mentioned about adeno-associated viral vector.
Right.
What do you mean by adeno-associated viral vector?
So the vector has been taken. See, I'm not a subject matter expert in that, so my explanation can be a little weird. Basically, the adenovirus vector plasmid has been taken as a vector. The gene of interest has been put into that, and that has been infected into the cells. Viruses are known to replicate only with the host cells, right? So when the gene of interest is put into that vesicle, so it's basically a vehicle, right? So how do you put the gene of interest into a cell? And how does that gene go into our chromosome?
So into the humans?
Yes, in the humans. Because eventually why we want to do this? To treat the disease in human, right?
Yes, sir.
Now, if you directly put the gene inside, the gene will be treated as a foreign particle and destroyed. Second, you require to amplify the gene. You need more number of copies of the gene because it is not going to be like one gene will target one particular site and attach to it. We have millions of cells, so millions of copies would be required, and those copies will not have a successful integration also. So you require some vesicle or some vessel by which it will get integrated inside. Viruses are known to do this very well, right? So viruses do penetrate our body. They go inside the cell. They put the chromosome inside. They burst the whole cell. They modify our chromosomes. So now what is done is that the virus has been made ineffective. Now it does not contain any of its own chromosome. It contains only the chromosome that we want. And we have put the gene of interest over there. That viruses grow inside the cell, the host cell, which is outside the body. Now that means the gene is amplified by virus growing because it will just multiply and make more and more copies. After that is done, the virus is broken and that plasmid is taken and that put into a humans.
Okay. So it's just like the gene of interest only.
Yes.
Okay, got it, sir. Thank you.
Right.
Good morning, sir.
Good morning.
Sir, you have mentioned about nano antibodies, basically where the camel has only the light chains. So I want
Yes.
to understand the applications of that and why, in what ways it is useful. I
See,
want to understand.
the
Yeah.
antibody that we normally make in human body, whatever antibodies are there, they're very big size. If you consider a size of 150 kd is almost 50 times bigger than insulin. Okay, just to have an imagination, insulin is the smallest protein because insulin has only a single chain. Insulin is just a single amino acid chain, while an antibody is a globular protein. So it's a very big molecule to produce. It's a very big molecule to produce by the host cell, to extract it outside the host cell, to purify it, and to inject it into humans. If somehow I am able to reduce the size of that antibody by 1/10 or even half, that will really improve my process efficiencies. Okay. Ultimately, the aim of the antibody is to go and do its job by attacking a particular site.
Thanks for the explanation, sir. And my second question is, what I understood is pharmacogenomics and targeted therapy is similar. So is there any difference between those, or it's similar?
Personalized means they can be given to one particular human being. You have to identify. If we have insulin, for example, it is not personalized, right? Insulin can be given to anyone. It is not necessary that you have to give to human A only or human B only. It is not produced for specific people. But in case of cell therapies, in case of gene therapies, in case of pharmacogenomics, they have to be targeted to a particular person. That's why they are very, very expensive, because they are not mass produced. They have to be given to that person also. They are similar in that fashion. Pharmacogenomics is a study, it is a database, it is not a product. Okay, but gene therapy is actual genetic engineering and a product. Pharmacogenomics, what it does, it has the data of each and every human, and that human, what are the genetic makeup of that person. So they would know from the database how that person will react. Okay, so on that basis of that data, they would modify the drug, or they will reduce or increase the dosage of the drug. While in cell and gene therapy, what you are doing is you are treating the patient. Say, for example, the patient has a cancer, and now we are talking about CAR T cell therapy. The T cells from the patient are removed. The cancer by biopsy cells are removed. The T cells are activated. They are made more potent. And they are exposed to this cancer cells, so they become very specifically potent against that particular cancer. Then the T cells are grown outside the human body and then put back inside the human body. So that way they are different from each other, but they both are personalized medicines.
Thank you.
Also, you mentioned regarding IgM antibodies and the IgM, right? IgG and IgM.
IgG and IgM, correct.
So, what is the like? What? How do we use it as a therapy?
It would be used similarly to IgG. See, IgG today, all anti-cancer drugs that we get, they are all IgGs only. The other examples of bispecific antibodies or conjugate antibodies, they are all mainly IgGs, because that is the
No, but
smallest
that
one.
is a long-term immunity base, no, sir. IgG.
That is when it is produced by human body. You are talking about active immunity. I am talking about passive, something which is given from outside. Okay. So you are giving antibodies, making them in bioreactor, and then giving them to humans. Why? Why are you doing that? Because you are not able to produce it. That human is not able to produce it. Either it does not have enough quantity, not exposed, or you don't want to be exposed to a disease to get it, right? So that's why we are producing it outside human body and giving it. So like camel antibodies are smallest, IgG is little bigger, and IgM is the biggest. But IgM has many sites. Because it has many sites, it can be used to attach to many different things. So just imagine that one particular cancer cannot be treated by one drug. It requires multiple drugs. Now, to carry those multiple drugs to a specific target, you can't use an IgG, because it has only one site which is empty. One site is going to attach to the cancer molecule. One site is empty. But an IgM has 9 sites empty out of the 10. So they can carry higher payloads or different payloads also. Possibilities are enormous, but the risk is also very high, because it's a very big molecule to produce, very big molecule to purify, and very big molecule to inject into any person. A higher protein leads to higher pain also. If you have taken some vaccines which were not clear, I think everybody has taken COVID vaccine, right? So COVID vaccine was not very clear. It was little turbid, and it was very painful also to certain people. It pained after a few days also. Why? Because the payload is very high. There is higher concentration of protein inside. The bigger the protein, the more the pain in injection. So probably we will have to go through IV route, not subcutaneous.
So IgG therapy is commonly used, no?
Yes, commonly used.
But it has only 2 binding sites.
Yes.
Hi, sir.
So, yeah.
Sir, you have spoken about this pancreas being replaced, or the stem cell and all that. Like, can you please explain a little more, because we are seeing many type 1 cases. Okay, are you talking about the stem cell being transferred and the islets of pancreas being activated?
Yes.
Because
Yes.
in
That's
India
a
we
very,
see
very
so
recent
many.
case. Very
Yeah.
recent example has happened. First
Yeah.
case, in fact, where the stem cells have been modified, they have been put inside the human pancreatic cells, they have converted into IL cells, and the patient seems to be cured of diabetes, because he has started producing his own insulin. And that is also in regulated fashion. There is a feedback loop which is happening. So this is the first case, and it's very promising. So yes, this is like a very common disease. A lot of patients, as you rightly said. So the impact
In
is
children,
huge.
especially in children, like in type 1, we see
It
that,
can be
right?
used for anybody, right? So
Even for type 2.
the
Okay.
possibility opens up for everyone.
Okay, okay.
The cost is a different matter altogether. Yes, that is going to be very costly initially, because main thing is, again, it is a personalized therapy, right? Because stem cells
Okay.
cannot be mass produced in a bioreactor. I cannot use
Okay.
my stems for you. You have to use your own stem cells for that purpose. That means the stem cells
Okay.
will have to be harvested from the body. They will have to be genetically modified. They will have to be grown, and then again they will have to be injected into the body.
Okay,
And they
into the
will have
same
to be injected in a
location.
particular organ of the body. So
Okay.
this makes it little difficult and costlier.
Okay, okay. Because we see many type 1 cases where they're using a lot of CGMs, monitoring, and doing it's pretty
Yes, yes.
expensive, all the
But
insulin and
that's
everything.
way cheaper for masses. This is something which is going to be probably a permanent treatment. The time only will tell. But probably a permanent treatment, I think it would be more effective to people who do not have any other way to go ahead.
Okay, okay.
But
Thank you,
sir,
sir.
even stem cells have limited capacity to grow, no, sir? Like
It
they
depends
don't
on the
remain stem
environment.
cells.
See, the knowledge about the stem cell is growing day by day, right? So initially, yes, we thought the stem cells are limited to certain things. We also thought that this is possible only at the stage of fetus, where the stem cells can actually divide into different types of organs and cells. But now it is made possible that we have been able to harvest stem cells from different organs of the body, not only from the bone marrow. And those stem cells have actually been reversed into the nascent stage, where they're pure stem cells and not converted into different type of cells. And now the study is going on where they are incubating these stem cells into different types of environments, different types of mediums, by which the stem cell differentiates into either a blood cell, or maybe a skin cell, or maybe a liver cell, and so on. Because that is happening naturally. Now we are trying to do this outside. So a lot of understanding and more data is coming out. So probably as time goes, it will become much better.
Thank you, sir.
Yeah.
Excuse me, sir.
Yeah.
So, as a general question, you told the novel biologics drug are related to cancer and other antibody diseases, autoimmune diseases. We all know, in Russia, they discovered a new vaccine. It will be a novel one, or it will be a new vaccine, trying for cancer, sir.
Okay. So as a question, or I don't know about the Russian vaccine, what is the new type? You can throw more light on that. I mean, there are many things going on. So which specifically are you talking about?
Like it will be novel drug, or it will be only specifically for only cancer cells,
I do not really know, dear, about what that Russian discovery is all about. As I said, there are many things going on. So if you can be more specific, then I can answer. I really don't know what the Russia technology is. Is it for a particular disease?
Yeah, so they have developed some vaccine for cancer to
cancer,
cure the
sir.
cancer. But sir, my doubt was I was also thinking, how can it be, like cancer? Cancer has all of different types: brain cancer, stomach
Yes,
cancer,
yes, yes.
pancreatic cancer. So how can one particular vaccine can treat all the type of cancer? Like in brain cancer, when we talk, there is a blood-brain barrier. So these all things, I think like a particular vaccine can't be treated for all the
Very good
cancer.
question. Actually, very good question. See, this is the actual problem with cancer. The term cancer is so commonly being used now that we all think it is one disease, but actually it is not. So it is like so many types of cancers are there, and each require different type of treatment. Some are easily localized, and some are like diffused and distributed throughout the body. So how will you attack this kind of cancers? Some are so deep inside the tissue that it is not possible to reach by injections. So various treatments are going on. If they have discovered a magical drug which is like treat all, fantastic. That would be like destroying every other market. But I really don't know. I have to study more on that. If you can send me the link across, I can tell you more about this. I really don't know. Okay, there is
Excuse
a
me, sir.
question here, sir. Does the immunogenicity of stem cells in the case of therapy vary from source from which it is produced? Is the efficacy the same? See, as I said, this is specific to person, and one person's stem cell cannot be given to another person. It is order locus. Now, if it is a homogeneous kind of a stem cell where it is non-specific and can be given, yes, studies going on there also. In those cases, immunogenicity question will come. But when a person's own stem cells are given, the immunogenicity question is not there because you are giving back their own cells. Okay, so we should not be producing any response to that. Okay, somebody wants to know about pharmacogenomics. So as I said, pharmacogenomics is nothing but a study by which they study the genetic makeup. There are certain markers which are known markers and how a person responds to a particular drug. I gave an example of chemotherapy over here. In case of chemotherapy, the same dose is given to different people and different people react differently. Some people react very badly. Some people do not have any symptoms. Why it is that way? Because they are different people having different genetic makeup. So pharmacogenomics is a study where they study the genetic makeup and the dose of the drug is given according to the genetic makeup. Okay, higher dose, different types, or a different way to be given. And that's how the pharmacogenomic study actually happens. So it is personalized medicine.
Customized medicine like that, pharmacogenomics?
Yes, it can be used for customized medicine, but mainly how it started was whatever medicines are already available can be delivered to individual patient in a customized way. Okay, so then it evolved into customization of medicine.
Okay, so
So does exosome research also, I mean, exosome treatment for cancer also come under personalized medicine, sir?
In exosomes, no, exosomes are not actually, they are just vesicles, right? So they are not personalized. But again, a lot of study are going on there. I don't think anything has been commercialized so far because it's very difficult to produce them. So, okay, one question is how does the plant ensure correct protein folding and post-translation modification like glycosylation? Yes. So here the plants have been identified that can do the job. Okay. Or they are genetically modified to do the job, right? And all the proteins cannot be made. So you cannot have mAbs for treating cancer being produced by plants because they are very complicated molecules. And such kind of high glycosylated proteins probably right now cannot be made in plants. Okay. So simple proteins would be made, like somebody is trying to make insulin in plants. Insulin is a very small molecule and can be produced in two different forms and then mix outside to make a different type of analog. And that those kind of simple molecules can be done. Like the example which was given about colicin. So that's a very small protein which requires very small amount of folding or maybe does not require any glycosylation. So it is specific to type of protein being used. Okay. Everything cannot be made so far. Okay, there's a question, can we harvest stem cells any age people or can collect stem cells at any time of born? Yes, initially the thought process was that it can be done only at the time of birth, and that's why many people used to take the cord blood cells and do the storage of cord blood cells because stem cells were supposed to be present. But now it has been known that yes, you can harvest stem cells from different organs also of the body from a aged person. Okay, what is the age, what is the health of the person, all those things need to be determined. Of course, better to be younger. As you grow old, the stem cell quantity reduces. Initial thought was we do not have any stem cells, but now the thought is that yes, we do have stem cells because there is a regeneration which is happening, and that is not possible without having stem cells. They are there, but they are hidden. So how do we know? How do we differentiate a stem cell from a normal cell? That's the whole research going on. So how to differentiate? Once you know how to differentiate, then you can harvest. How do vaccines interact with other medication? Okay, and health conditions. I've given example of mRNA vaccine for COVID. I think if you can go back to that and see, basically vaccines are very different types of molecules which actually bring about an immune response in human. So this is called passive activation. Why? Because we are giving an antigen which is not potent to reproduce. A virus or a bacteria which can grow inside a human body will create a disease because it grows faster than our immune system's response. Right? That's why we get disease. If the growth is not fast, then our immune system is able to tackle that and bring it back, okay, to normalcy. Say, for example, your normal cold or flu that you get, you have symptoms for a few days, our immune system is able to trap that cells and destroy them. Maybe very few people have to take medication. But in case of vaccines, where suppose you have poliovirus grows very fast once it attacks and goes inside the cell. It is difficult for an immune cell to go, it cannot go inside our cells, right? It can only destroy what is outside the cell. Now, poliovirus is inside the cell. So you can't have your own immune system doing that job, and that's why you have to give vaccine. So vaccines is something which is ready-made solution, which is available, goes out and destroys or neutralizes all the polioviruses, and that's why you are not infected by them. Is there any side effects of novel biologic drugs? Yes, there always will be, right? So because everything is not known, it's a very new drug. Yes, definitely it will have. Yes, it will have autoimmune disease like symptoms also. It can have. What is written is, yes, it can create autoimmune kind of reaction also. And that's how we study immunogenicity before giving the injection to market first by doing clinical studies and studying this kind of responses in human. Can gene therapy be used to treat cancer? Yes, they are trying to do that. But difficult, more or less, it is more on the cancer. If related to a gene mutation, then yes. If it is not related to gene mutation, no. How they can reduce side effects in genetical diseases? Well, more and more amount of studies, human clinical trials have to be done. More human genes, more human proteins have to be there. Like right now, there are chimeras. In chimeras, matlab, you have some amount of animal antibody, you have some amount of human antibody, which is fused together, and those are given. And when those are given, they create that immune response. So more and more amount of people are trying to get completely human antibodies. So that can be replacement for those. So all of you have a very nice day. Thank you very much for attending and being a very good audience. Thank you for your questions. They were very, very, very nice.